Great. Well, thank you. Thank you so much to the organizers for having me here, and thank you all of you for such a great conference. I've been really blown away by the talks, and I'm looking forward to the upcoming talks and also to look at some of the posters that I haven't seen yet. Yeah, this has really been an incredible day so far, and keep it up. So, all right. So since my days making gold, now I have to figure out how to use all that money. So that's what I'm going to talk to you about today. So origami movement microscopy is a technology I invented as a postdoc in Xiaowei Zhuang's lab at Harvard, and that's a big part of what we do in my lab today. But we also work on some other projects that in some way relate to biomechanics. So I'll actually take you on a little bit of a tour through kind of the things that we're excited about, and I hope that you'll get something out of it and that maybe it inspires some of your work in the future. All right, so first, just to get us oriented, so where is Salk? So this is where we are now, and I'm down here. So it's basically like here, but less mountains, better weather. This is what it looks like. We're in this beautiful building that is built on the cliffs above the Pacific Ocean, and it was designed by a famous architect, Louis Kahn. So actually this channel here in the middle is aligned with sunset during equinox. So twice a year we all gather here and we see the sun set over the ocean, and we feel like people building the Stonehenge or something. All right, so I like to show this video to begin with because in the fraction of a second that you just saw pass here, this picture has accelerated this little artificial rock from a speed of 0 miles per hour or kilometers per hour to a speed of 100 miles per hour or 160 kilometers per hour. And it's really, when you think about it, the more you think about it, the more of an incredible feat it is, really. And this ability of humans to throw rocks and other projectiles at these speeds has most certainly driven our evolution as a species. And I think that illustrates very well what an incredible mechanical device our human body is. And so just like any mechanical device, it consists of moving parts. But what's different about our bodies compared to other machines is that we have moving parts on all scales, from the muscles that contract to give us this movement to the proteins inside those muscles that that generate the force that then propels the movement of the muscles, and all the way to the enzymes that are reading and maintaining the DNA to give rise to the proteins that then give rise to the forces. So on all these scales, we have movement and mechanics, and it's only by all of these different components working together in unison that we can have a functioning body. So in my lab, we're focused on understanding how biomechanics across these scales give rise to function in biology. And today, I'll tell you about 3 different scales of investigation. So first, I'll talk to you about this method I invented, origami movement microscopy. And then I'll take us up to a higher level where we use force spectroscopy to understand the elasticity of muscles. And finally, I'll give you a little taste of what we're working on to understand heart disease and specifically how to use fluorescent methods to see gene expression across tissue. All right, so let's get started with origami movement microscopy. This all came about because I was really curious about what DNA is up to when you're not looking. So organs consist of cells. Cells consist of, most of them have nuclei, and the nuclei contain chromatin. Chromatin is DNA and proteins and a bunch of other stuff, but basically DNA and proteins. And if you zoom in on what the DNA looks like, it's not, in eukaryotic cells, it's not like a flexible polymer. It's very much twisted and torqued and bent into weird shapes by lots of different proteins in all processes that you can imagine. So there are enzymes that read the DNA, there are enzymes that replicate the DNA, there are enzymes that twist DNA, block DNA, sequester DNA, and so forth. And so for each one of these reactions, there's a mechanical movement associated with it. But for most of these reactions, these movements have never been seen, because as you can imagine, these are very small movements, and it's very challenging to capture them. So that's what I set out to do, just like many of you are doing very well. And the way I thought about it was to decompose the movement into separate modes of separate degrees of freedom. So if an enzyme or a protein is interacting with a piece of DNA, then the DNA can move in one of several ways. So 3 of the main modes of movement are rotation, linear motion, or bending. And this is true for any protein interacting with DNA or any small molecule interacting with DNA for that matter. So if you have transcription, this enzyme is moving along the DNA, but from the point of view of the enzyme, the DNA is moving. In reality, it's a bit of both, depends on which side you're on. That's true for enzymes that remodel chromatin and enzymes that edit genes as well. So in our lab, we're currently studying these 3 systems, and we're developing methods to study all these modes of movement. And how do we do that? Well, I'll get to that in a second. First of all, let's focus on rotation. Rotation is a very convenient mode of movement to study because anything that moves along the axis of DNA will also cause rotation because DNA is a helix. And so from the point of view of an enzyme such as RNA polymerase, when it's moving along the DNA, it basically pulls the DNA into itself, and then as it's pulling it in, it's also twisting it because it has to follow the axis of the coordinate system of the bases. So many DNA processing reactions generate rotation, and if we can track the rotation, then we can follow these reactions in real time. I'm not the first one to think of this. Actually, I was inspired by this legendary paper from Kinoshita's lab in 2001, where he wanted to know if RNA polymerase in fact did rotate DNA as it walked on it. And so to see it, he attached a magnetic bead to the end of the DNA, and then he attached little fluorescent beads to the magnetic bead, and then he used the magnet to hold the magnetic bead upright, and then as RNA polymerase that was tethered to a surface, as it was transcribing this DNA, he watched for a rotation in this magnetic bead. And he actually captured these beautiful trajectories where you can see many, many, many turns of rotation as over the course of time. And what he saw was that at different concentrations of fuel or nucleotides, he saw different speeds of rotation. But as you can tell, each enzyme is slightly different, and each reaction follows a different trajectory. And in the final paragraph of this paper, he said that, well, we expect that the RNA polymerase rotation reported here could provide a means of resolving individual steps of transcription. Maybe we can actually one day see these single steps of movement as this enzyme reads the DNA. Transcription of one base will produce a linear translocation of 1/3 of a nanometer. So the distance that this enzyme moves from one base to the next is very small, difficult to see, especially in a Brownian bath. And the rotation that is associated with this movement is 35 degrees, which is also very small, but it should be detectable, especially if you can amplify the rotation like this. So as Kinoshita correctly stated, the main problem here is torsional Brownian motion of DNA, which has to be averaged out. So to put this in clear speech, what this means is that even if there's no rotation happening down here, because there's a lot of Brownian dynamics, this bead on the top will swivel around randomly, and you have to average that out to see exactly what's going on. So the amount of Brownian motion that happens here depends on a few different parameters. The main ones are drag, hydrodynamic drag, and the stiffness of this entire contraption. So a tag much smaller than the bead and shorter DNA is needed for averaging within a reasonable time. All right, so this seems like a system we could optimize. We can do better than giant magnetic beads in our day and age. So if we look at the equations that govern the resolution, we can get the resolution in space or in angle. That's given by, so essentially, if you take an instantaneous picture of this and you see exactly what angle the bead is in, you get some angle. How well does that angle reflect what's happening down here? Well, it reflects it down to a precision that is, or an accuracy that is given by this equation. So as you can tell, the stiffness, if you have a higher stiffness of this entire contraption, then you have a smaller standard deviation, so you can more accurately determine the angle. But then there's also the time dimension, because you can always average this for a very, very long time, and then you get the actual angle. But how fast the angle averages out to a smaller, to a higher accuracy depends on both the drag and the stiffness. So again, if you have a higher stiffness, you will have a much better resolution in time. Like basically, you have to average for a shorter time to get the angle you want. And if you can minimize the hydrodynamic drag, this resolution gets better as well. All right, so to improve resolution, we need high stiffness and low drag. So let's design something that has high stiffness and low drag and amplifies the rotation of DNA. So the way I thought about it is, if we could just attach a lever arm on top of the DNA, that would serve the purpose, right? Like, if there's something fluorescent here, even better. We can see it rotate. So if this lever arm is about 80 nanometers, then a 1 base pair shift should correspond to 40 nanometers, which we should be able to detect in a regular fluorescence microscope. So this lever arm has to be rigid, it has to be sleek, and it has to be easy to connect to DNA. And that led me to the technology called DNA Origami. So let me give you a quick overview of 40 years of development of DNA Origami. It started with a paper in the Journal of Theoretical Biology in 1982 by Nadrian Seeman. He basically had the idea that if we can decide the sequence of DNA, that we can get different single strands of DNA to combine into custom structures of our choosing. It took until 2006 before this was sort of put into practice in a useful way. And so Paul Rothman at Caltech, he had this idea that if you take a scaffold DNA, so this is single-stranded DNA from a virus, for instance, it's something with a given sequence, and then you synthesize a bunch of staple strands. These are short sequences of single-stranded DNA that can base pair with the scaffold to pinch and pull the scaffold into a certain shape. If we do that in a clever way, then we can get the scaffold to go into basically any shape we want. So this size is limited by the size of the scaffold DNA. Typically we use a viral scaffold, and it gives us about 100 nanometer structures. And so in the first paper he showed you can route it in different ways, and this creates actual shapes that you can then see with AFM. And since then there's been a lot of advancements to make this into 3 dimensions, to make curved shapes, to make wireframe shapes, and to even make higher order assemblies that approach the size of cells. So now we have this technology that we can build anything we want on a size scale from nanometers to microns. So let's build a DNA origami rotor. All right, so I teamed up with Peng Yin, who is a professor at the Wyss Institute at Harvard Medical School, and his graduate student Ming Jie, and we built this rotor. Basically, it consists of 200 pieces of DNA. Half of them are half of the bases are synthesized, half of them are part of the scaffold that comes from a virus. And then a couple of these staple strands here are labeled with fluorescent oligos. You can just order them from any DNA synthesis company. So this should allow us to see this rotor if it's rotating. This is what we thought about what it would look like. So if we have an enzyme that moves on DNA, we attach it to a microscope slide, and then we feed it with DNA. If it moves along the DNA should move down and also spin, and that should rotate the propeller or rotor, and then we should see that. All right, so we made these rotors. This is what they look like. Here's a zoomed in version of potentially the smallest rotor ever made. I don't want to, that might not be true, but smallest propeller I've seen at least. All right, so nothing to compete about anyways. So first we wanted to try it on something simple. So we took a helicase enzyme, RecBCD. What it does is it basically eats DNA. It's an enzyme that is found in bacteria, and it responds to DNA damage. So if you stay out in the sun, not a problem in Vancouver, but in San Diego for sure, then you'll get damage to your DNA, and that could cut the strands of DNA. So then RecBCD, or the equivalent enzyme in humans, will jump in, attach to the end of the DNA, and then just like basically chew it up until it hits a certain spot from which it starts to repair it. And the reason it can repair it is because we have 2 copies of every chromosome. Bacteria sometimes do too, so when they're dividing. Anyway, so this seems like an easy protein to work with. It chews up DNA, and since it's like eating base by base, it basically pulls the strands apart, and so that should cause the DNA to spin, we thought. Nobody had seen this rotation. It's very fast. It actually unwinds up to 1,000 base pairs per second. And the way it works is basically, this is the enzyme, here's the DNA. It has 2 motor domains. Each one grabs onto a single strand, and then it pulls those strands. In the presence of ATP, you can pull those strands in different directions across a pin. That should spin the DNA. All right, so this is how we did it. We took a microscope slide. We added a bunch of RecBCD and buffer. So proteins stick to glass, so they just sit there. And then we fed this protein with a bunch of DNA rotors with double-stranded DNA that had a break. And so if RecBCD worked the way we thought it would grab onto the end here and then start unwinding. And so in the presence of ATP, we should see the rotor spin. So since this works so well in PowerPoint, we thought, might as well try it in reality. Here's what you see in a microscope. Each green spot is one rotor. You can see the tip one of the rotor blades is fluorescently labeled. If we zoom in on one of these spots and we add ATP, this is what it looks like. These are pixels on the camera. This is at 500 frames per second. And you can see that maybe it's wiggling. It was like slightly underwhelming when we saw this. But then we realized that these rotors are about 150 nanometers, which is about the size of a pixel. So we wouldn't expect it to go in big circles. We would expect it to go in small circles. In fact, the circle should be about the size of a pixel. Fortunately for us, there's only one rotor here, so we can fit this distribution of light to a 2D Gaussian, basically an approximation of the point spread function. And that can give us a very specific coordinate where the center of fluorescence is for each frame. And then we can reconstruct this trajectory that is more zoomed in. And what we see is this is the same trajectory as here, but here we've fit this distribution with the centroid. And so you can see that it goes in circles, and the size of this circle is about the size of the rotor. So this gave us confidence that what we're seeing is actually the rotation of a single rotor, and it seems relatively unidirectional. There's a bit of wiggling, that's mostly Brownian motion, but there's definitely a directional movement as well. So we can take this trajectory and then plot it as a rotational angle as a function of time. But since we know this 35 degrees per base, we can actually tell how many bases this enzyme has moved along on the DNA, and we can get a reaction speed from this. If we plot the speed of reaction, so like the slope of this thing as a function of ATP, we can see that with more ATP, it's faster. We can fit this to Michele's mental model and see that it actually matches what people have found in bulk as well. So what we're seeing here is very accurately reproducing what's happening in cells. What's really powerful about this method is that not only can we get a single molecule trajectory, but we can actually get it for hundreds to even thousands of trajectories in one field of view. And we don't really have to do anything. We just put these things there and then watch it, right? So we can get massive amounts of data with very little effort. This experiment took about 2 minutes to set up. So it's a great project for grad students, if you're interested. And what pops out is that there are no enzymes that are similar. I mean, they each do their own thing. Some of them, they unwind the DNA and then they go backwards, and then they hang out for a while and then they keep going again. And some of them just go straight away and they have somewhere to be, right? So now we're at this stage where we're sort of going out in the jungle and observing the animals in their own habitat. And so we're trying to figure out what's actually going on here. If you want to learn more about what we found out for RecPCD, you can read the paper. We thought, well, now that we have this capability of seeing rotation at such high resolution, maybe we have good enough resolution that we can capture the single base pair snapping of RNA polymerase. Maybe we can actually see some more details that people haven't seen before. And to get there, we first wanted to quantify to see exactly how good is the resolution. So we took our rotors and we anchored them to a microscope slide using this origami tripod thingy that is basically just a rigid anchor that keeps it oriented in the right way. And then we looked at how much it fluctuates and moves without anything rotating it. So this is just a reflection of the Brownian motion. And as you can see, it rotates quite a bit, back and forth, back and forth, back and forth, back and forth. How much depends on how much flexibility you have in the DNA here in between. We can also average this trajectory and then get a higher precision or accuracy in our measurement. And this is exactly what we would expect. If you average more, you average out the Brownian motion, you get more accurate detection, but at the cost of time resolution. We can take a Fourier spectrum of this and get the power spectrum. And here's it's fit to a Lorentzian. What you see here is at different frequencies, you have different amplitude of movement. And you can see that there's a cutoff frequency around a couple of hundred hertz. Above this, there's a lot of damping. So this tells us that any movements that are faster than about 100 hertz are going to be heavily damped by the drag of the situation. And another way to depict our time resolution is to look at what's the standard deviation for a chunk of this trace as a function of how long we integrate. So if we average for a longer time, we get lower standard deviation, meaning higher resolution. And so a single base pair rotation would be about 35 degrees. So if we want a signal to noise of 1, we would have to average for about 1 millisecond. If we want a signal to noise of 3, then we need to average for about 20 milliseconds. So what this tells us is that we can detect a 1 base pair rotation with a signal to noise of 3 at 50 hertz. And this is about a 1,000-fold improvement over the paper from 2001. It was already very impressive. So with these results, we were like, okay, let's see. Can we see these steps? Does RNA polymerase, in fact, rotate DNA in steps? Well, again, it worked in PowerPoint, and it worked in reality. See a trend here. So these are the pixels again. Here you see the trajectory of the rotor, Maybe you can already see some stepping. I'll just put it in context. Here's the data from Keynote. Here's our data. You can zoom in to a tiny bit. So these are entire revolutions, right? This is like 50 turns. And this is, meanwhile, this is 5 turns. So we can, and you can see the time axis is totally different here too. This is like an hour, and this is 30 seconds. So we can get much higher time resolution, much better angle resolution, and if you zoom in even more in low concentration of nucleotides, you can start to see these single steps that are surprisingly similar to the shift between base pairs. And in fact, we can take these trajectories, fit them with a Hindenburg model that is a bit of a questionable approach, but if you do it right, then it can actually give you useful information. So when we did this is sort of an unbiased way of figuring out if there are steps in the data. What it came out was that there was a distribution of step sizes with a peak around 35 degrees, which you might remember. So we can conclude that this seems surprisingly similar to what we would expect. Probably it means that RNA polymerase takes single base pair steps. But what also stood out to us was that these steps were actually not the same height, all of them. In fact, it seems like there was some difference in step size depending on what sequence there was. So maybe now we're speculating that maybe from these trajectories, we could actually tell what the sequence of DNA is. And this would be at present time, it seems like a cumbersome way of sequencing DNA, but we did the math, and this is actually cheaper per base than any other sequencing method currently available. So maybe that's an application we can do. We're not consuming anything really in these experiments. We could also potentially see from these trajectories if there are any modifications to DNA or if something is stopping the movement of the enzyme. So now we're using this to figure out how transcription is controlled. All right, so we're also using this method to measure these other movements, linear movement and bending, and we're just trying to be creative. And I encourage all of you to think about, like, if you had the ability to build anything on the nanometer scale, how would you visualize these other kinds of movements? And we're using these technologies to study different forms of proteins interacting with DNA. Our goal is to visualize all of these movements and create assays for other people to use. And I want to leave you with this message that with origami movement microscopy, we can amplify and visualize molecular motion. So just like the molecules, we need to move a little, move around in your seat, take a break, get ready for part 2. All right, cool. Good. I think everyone should have this. Like, yeah. Not the other talks were fantastic, but people who have to torture their audience for 45 minutes definitely need to put some breaks in. Question?
Just a quick question. Why the cross geometry instead of just a single rod?
Haha. That's a great question. Can you answer? Well, I was just, at the time, I was a naive postdoc, and I was like, oh, actually, the way this all came about was I talked to my advisor, and I was like, I really want to, I just feel like there must be a better way to measure rotation than these humongous beads like wobbling around, and you have to attach all these different things to one another. And she was like, well, don't tell me there are better ways, like tell me how to do it. And so I was like, okay, fine. And I went back to my desk, and on my desk there were 2 pens that were lying like this. And so I was like, the light bulb went off. And that is the honest reason why there's the cross. But then I thought about it, and I was like, okay, can I just do it with one? But the more I thought about it, the more I was like, well, we can't really control the orientation that the DNA is coming out of the enzyme. And so if it's just one bar, maybe it flops over on the side, and maybe it's not stable. So I felt like with having these crossbars, at least it would ensure that the orientation is held perpendicular to the slide. And in fact, the aspect ratio is such that the sort of rotor blades are longer than the stem of the DNA. So that imposes a certain geometry on the entire setup. And turned out, years later, that was correct. I found out that was correct, but that was like, just, yeah.
It's like ATPase is measured with a tubule, right?
Right. But the difference there was that when Keynote measured the rotation of F1 ATPase, so the study is referring to is there's a rotary motor that produces ATP, or it can use ATP to create the proton gradient. But it also has a stator and a rotor, and the way he set up that experiment was he oriented the motor itself, so the rotational part was perpendicular to the slide. I didn't have that luxury, so I used this strategy instead. In fact, I think this strategy could be used to orient the way we attach things to surfaces in general. We're exploring that as a method could be used for in biotechnology. But for now, that was the, it actually, for some system, it works perfectly fine with a single rotor. Anyway, I got to move on. I'll take more questions afterwards. But do feel free, if it's something that really bothers you, just do shout out. I'm happy to take intermittent questions. Otherwise, save to the end. All right, part 2 is force spectroscopy to understand muscle elasticity. Let's get back to this video. I just love this video. It's incredible. If you look at it, there's something very strange about it. Oh, there are many things that are strange. This is one of the things that are strange. It turns out this pose is absolutely essential for pitchers to generate the kinds of losses they do. Can anyone tell me, why does this look strange? What's strange about this? Try to do it. Try to do it. Can you do it? This is like, it's almost 90 degrees backwards, from his angle of his body. I can't do it, yeah. Turns out he can't do it either if he's standing still. The reason his arm is in this direction is because he's loading the muscles in his shoulder and his pectorals in such a way that they're extended beyond their normal range of motion. And what that does is it spring loads a bunch of components in your muscle system so that all of that kinetic energy from like rotating your body gets converted into potential energy, and then in one fell swoop he can harness all of that potential energy, recoil all the springs, and in fact that recoil that comes from all of these torsional muscles contracting that swivels around his arm, the speed of contractions is much faster than what you could achieve with ATP-driven contraction of myosin actin. Okay, that was a little bit of a spoiler. Turns out this has very little to do with myosin actin, much more with passive elasticity of muscles. Passive elasticity is actually critical when you throw something, when you jump, or when you run. Take another example. If you run a marathon, every time you take a step, you contract some muscles and you stretch out some other muscles. So when you step down, you're actually extending these muscles on the back of your leg, and your Achilles tendon is under a lot of force. And then when you kick off, you have to contract those muscles. So at first you extend these muscles under force, and then they contract again. So you go through these cycles where you basically step down and extend the muscles, and then you contract them to kick off, and you extend and you contract. If you had to push yourself off the ground for every single step for an entire marathon, you would have to eat, I don't know, like a zoo or a forest if you're a vegetarian. It's a lot of energy. Turns out that when you run, it's much better if you could just bounce. Like a pogo stick or a bouncy ball. If you throw it, it will just bounce. It doesn't have to use any energy to bounce. It doesn't actually, consume any chemical source of energy. It's just converting potential energy into kinetic energy and vice versa. So if we can do this, and this turns out to be a big difference between mediocre runners and great runners. Runners who can run for a marathon with using very minimal ATP are the best marathon runners. So it's really good to learn where the muscle elasticity comes from and how we can use it best. It also turns out that it's very important for your heart to be able to convert kinetic energy into potential energy. Because every time your left ventricle fills up with blood, it stretches from the pressure of your internal blood pressure. And that pressure swells up the chamber to a certain size. And then when you contract your chamber to pump out blood, all of that energy has to be converted into movement of blood again. So if that is not an elastic process, then you have to use a lot of energy to create the same kind of pumping. And it turns out that if you have a heart attack, then part of your heart wall essentially dies and gets filled with scar tissue, and that is not as, first of all, it's not as elastic, and it's also not as, it's stiffer than what the muscle tissue is. And so you're less efficient at converting the swelling energy into recoil. So we would like to understand the molecular origins of muscle elasticity, and maybe we could even change elasticity. If we could do that, then we could both be better baseball pitchers, and we can maybe be healthier people overall. So where does the elasticity come from? Well, let's take a look at the muscle. If you zoom into a fiber, this is true for both skeletal and cardiac muscle. There are a bunch of fibers. These fibers consist of sarcomeres. The sarcomeres are the fundamental contractile units in muscles. Sarcomeres consist of a bunch of protein elements. The three main ones are myosin, actin, and titin. You've already heard a little bit about titin. Turns out titin is the hero of this story as well. Titin sets the elasticity of the muscle. In fact, if you cut titin, then you lose about 70% of the stiffness of muscle tissue. So most of the force in muscle is held across titin if it's relaxed. Titin looks like a knotted rope. There are little domains of proteins that are folded independently. And yeah, let's see what happens if you pull on it. So we used an AFM. This is a way to apply a calibrated force to a protein. The way we do that is we have a movable stage that we can move down. And when it moves down, it pulls the protein pulls the cantilever bends, and that deflects the laser to a different location, so we can use this signal to detect the force. We can then build a feedback system that allows us to basically pull it until it achieves a constant force. And then we just hold it at that force and see what happens. And if something gets longer, we just pull it until it reaches the same force again. So we built a microscope like this that is now commercially available. If you want to do this experiment at home, you just have to pay some money to this company, and then you can get your very own. And with this microscope, we were able to pull single Titin molecules, as well as other people have done this. But what really strikes me is that if you measure the length from here to here, and here we have, this is 5 domains of Titin. Each domain is about 4 nanometers, so the total length from end to end should be about 20 nanometers if it's fully stretched out. Okay, not quite 20, but it's like 50 nanometers or something. So here's time. We're applying a pretty high force, and over time, you would expect it to just go to this length and then stay here, right? But this is what happens. So over time, you see these like steps, and we figured out, no, we didn't figure out, other people figured out before me, that these steps all correspond to individual domains unfolding. So this happens in muscle as well. When you apply a force, here's the cantilever tip, here's the surface. If you apply a force to Titin, or to any protein for that matter, if the force is high enough, these domains will unfold and release a lot of polypeptide. And this can do a lot of things. It could be a protective mechanism. Maybe you want to make the muscle longer without like breaking anything. It also turns out that this mechanism is completely reversible. So if you unfold these domains and then you remove the force, then it collapses back in kind of a single motion. But then if you pull again, you see these steps again. And these steps actually indicate to us that these domains have reacquired their native fold. So it's a reversible process. It can go from long to short, okay? So we were like, maybe this has something to do with why the muscles behave differently in different parts of your body and why it behaves differently at different times and under different conditions. In fact, if we can modulate this balance in some way between unfolded and folded, that would explain why muscles are sometimes soft, why they're sometimes stiff, and why they're sometimes more elastic than others. So we looked for candidate mechanisms to modulate this folding, and it turns out that when you exercise, there's a lot of oxidative stress. So here's a marker for oxidative stress, and this is muscle tissue from human subjects before and after exercise. You see there's a lot more oxidation. The main redox buffer in the cells consists of this small molecule called glutathione that has a sulfur, or a thiol. And this thiol can allow this small molecule to attach to proteins. So if proteins have free thiols, which a lot of proteins do in the cytosol of cells, and you're under conditions of oxidative stress, then chances are you will attach one of these glutathione molecules to the protein. Haha, now there's a mechanism by which exercise can induce the attachment of a molecule to a protein. Maybe this is something that can be useful. In fact, we found that Titin is the target for glutathionylation in this Western blot. And we found that Titin has a lot of cysteines. And if you look at the sequence of Titin, most of the cysteines tend to be, or seem to be clustered here in this region that is actually the elastic part of Titin, the I-band that you heard about before. So now we have a lot of potential sites for modification. We have a mechanism for modification. But what we found was that if you look at the structure of these protein domains, most of these systems are buried, meaning that they're like inside the protein, so they shouldn't be accessible. But remember, if you apply a force to Titin, it unfolds. So this should expose sites. So now we're like, oh, wow, if you apply force to muscle, you might unfold Titin. And inside Titin, there are a lot of these residues that are susceptible to oxidation. And when you exercise, you generate a lot of oxidative compounds, and so you will increase the probability that something gets oxidized. Well, if it becomes oxidized, will that change the folding-unfolding equilibrium? Seemed like a no-brainer, but it was a little bit trickier. So we pulled on a single Titan molecule. This one has, I think, 8 domains. You see all these steps. Each one of these 8 domains are folded. We did this also in the presence of an oxidizing buffer, and actually we still saw these steps of unfolding, which means that it doesn't change the structure of Titan just normally. So an oxidizing buffer doesn't affect folded Titan. And then we exposed it to an oxidizing buffer for 2 seconds, and then we removed the force, allowed it to collapse again. We waited for 5 seconds, and then we pulled again. Oh dear, okay, so they refolded. So it basically looks the same here and here, meaning that there was no change after exposure to an oxidation buffer. And then somebody had the brilliant idea, well, why don't we just wait a little bit longer? And so we did the same experiment and waited a little bit longer, and now there was no refolding. So when we pulled the second time, this muscle protein was super compliant. It just went to the full length right away, no steps. And what that means is that the protein didn't refold into its native state anymore when it had been oxidized. So we found that this was a time-dependent effect. So this is how many % of the proteins refolded as a function of exposure to an oxidizing buffer. And we saw that the refolding dropped a lot. So this means that the muscle is more compliant with oxidation. And if we remove the cysteines, we didn't see this effect. If we had a reducing buffer, we didn't see this effect. And if we didn't use glutathione, we didn't see this effect. We then measured the rates, and we put together a kinetic model to explain, or to try to predict what effect this would have in an actual muscle, because we know roughly how many titans we have in an actual muscle, and we know roughly what forces they experience. And so this is simulating a heart over a time scale of hours. How much do we expect the stiffness to change for each titan within that heart? Well, it turns out we can reach the full sort of, from normal stiffness to a very low stiffness. This is all qualitative for now. But what we saw was that the difference between like the low stiffness and high stiffness happened in the physiological range of oxidation that we actually see in muscle. So this could potentially be a mechanism by which muscle elasticity is regulated. So then we took muscle cells and we exposed them to different buffers, from reducing buffers to oxidizing buffers. We saw how much force they generated when we pulled them to certain lengths. And we could see that with a reducing buffer, they generate a lot of resistive force, and in oxidizing buffer, they don't generate much resistive force. So these muscles are more compliant than these ones, and it's fully reversible. So the conclusion is that if you stretch under oxidative stress conditions, then that might modify titin in your muscles to make it softer. But if one of these two ingredients are missing, either the stretching or the oxidation, then you will not see the same effect. So the intuitive conclusion here is that like if you want to stretch, you better be warmed up and like exercise, so you have a little bit of oxidative stress. It's really surprising to me. Like after we found all this, I looked into literature, and like nobody really knows what happens in your muscle when you stretch. But I think that this mechanism can potentially answer why we can modify the stiffness of our muscles. So some people need to use high power, stiff muscles and short range of motion. Other people benefit from having flexible muscles that can work over a larger range of motion. And we know that we can go between these two with the right training regimen. This glutathionylation could potentially be one of the candidate regulatory mechanisms that can allow this to happen. And we could potentially use this mechanism as well to modify elasticity to cure some muscle disorders, including heart disease. This is something we're looking into now. All right. Stretching break. Get ready for the third part. Okay.
We haven't been exercising.
Yeah. Well, you exercised your brain. That probably generated a lot of oxidative stress. What is it? 60% of your energy consumption in your brain. So, all right. Good. So, in the third and last part of my talk, let's take another step up in scale and go to entire muscles. And to look at entire muscles, we use a new method called MERFISH, which is a way to spatially profile cells, or gene expression. So in this part of the talk, I'll first convince you that spatial profiling can reveal a really important new biology. I'll tell you how MERFISH works, and then I'll show you a little bit of example data from our lab that we got just in the last week or two while I was conveniently traveling. So here's a picture of a healthy human heart. This is from our collaborators at Harvard. They got their hands on some donor patient tissue from. So when people die and they're very kind to others, they can sign up to be organ donors, and that means that organs can be used as transplant organs for people who need transplants. Now, an organ is only good for a very short amount of time after a person dies. And so you have a limited time frame where you can match a donor with a recipient. And in some cases, you can't match it with a recipient, or the recipient dies before the organ can make it there. And then we don't have to waste that organ. We can actually use it to study it. So this is an example of a healthy human heart that was destined for a recipient. The recipient passed away, and so now we can look at it. So what you see in this healthy human heart with a trichrome stain is red shows you muscle tissue, blue shows you collagen, which some of you know a lot about in the audience. And so you can see that there's a lot of collagen in little clusters here. These are in fact blood vessels that are a little bit collapsed because this is a thin section of the tissue, so there's no blood here. But you can see these two blood vessels are reinforced with collagen. There's a tiny bit of collagen in between the muscle cells, but not very much. This could be like scar tissue or something, but it's not very prominent. Now, if we look at the heart from a person who is suffering from heart failure, a progressive heart disease that actually, I think 60 million people are currently suffering from, this is what a heart with heart failure looks like. So this person was, a functional human being, but in need of a heart transplant. And this patient actually did get a heart transplant, and it's now okay-ish. But you can see that the heart looks very different from a structural point of view. You see a large swaths of blue. This shows you that there's collagen and potentially scar tissue in between the muscle cells. So the heart is a mechanical organ. It needs to pump. It needs to produce force. It's not far-fetched to think that the structure of the heart, the structure of the tissue, contributes a lot to its function. And that is exactly what we want to figure out. We want to figure out, how does the cellular architecture lead to the function of the heart? Surprisingly, to me at least surprisingly, we don't really know what the structure of the heart is on a cellular level. I mean, if you look at this picture here, you see that these are cells, but it's very difficult to tell what kind of cells there are. We know there are dozens at least of different kinds of cells, and that's just by one classification. There are tons of different cells in here, but they look like blobs, right? We can stain one or a few of them at a time, but it's very difficult to get an idea of what all of them are. So that's what we want to figure out. We want to figure out what kinds of cells are there in a heart. How are these cells organized in space? And what are the microenvironments of the cells? Or from the point of view of the cell. So as a single molecule biophysicist, I always think from the point of view of the thing I'm studying. So from a cell's point of view, who am I? Where am I? And who are my neighbors? Good questions to ask yourself in general, I would say. All right. To answer what kinds of cells there are, we do have a great method for this. There is a method called single-cell RNA sequencing. What that allows you to do is to take a piece of tissue, dissociate the cells, put them through a pipeline that is now commercially available, and then read out the transcriptome, the genes that are actively expressed in each cell. And then for each cell, we get a readout of thousands of different genes and the expression level of each one of those different genes. And that gives you this like super high dimensional data set. And the way people typically visualize this is with these cluster plots. So these are low dimensional projections of that high dimensional data set. Each cluster, so each spot is one cell. This is from a human heart. And the distance from one cell to another in this space is defined by the difference in transcription. So you can see that blood vessel cells clustered together, muscle cells clustered together, immune cells clustered together, neurons clustered together, and so forth. So this gives us actually a good idea of what kinds of cells there are in the heart. But it doesn't give us an idea of where they are, because we have to separate them from the tissue. How are these cells organized in space? Well, what we really want is to take one of these cluster maps. This is for a mouse brain, but each one of these clusters is each spot is one cell, each cluster is one type of cell, one kind of cell. We want to take these clusters and we want to map them onto the tissue. We want to see where the cells are. As you can imagine, if we have this kind of map, this will give us much more information. And then we can start to see which cells are next to other cells as well. What are the microenvironments? The reason why this is important will soon become clear. Your neighbors actively affect your health. I captured this cell in the wild, and this cell happened to have a neighbor who gave him some nutrients, maybe even some antioxidants here. And so now this cell is healthy, well sustained. This happens in your bodies all the time. Neighbors help out. But the same cell could have a bad neighbor, This might be a natural killer cell or an immune cell that tells the cell to, die because it's infected, or maybe mistakenly. Autoimmune disorders are caused by this. So your neighbors affect your health. It's really useful to see which cells are next to which other cells. And if we can make these maps of cells, so this is in the human brain. This is actually in collaboration with Visgen, which is a spin-off company from my postdoc lab. You can see, you can see, different these are cell bodies in the human brain, and you can see that the color corresponds to the transcription profile of the kind of cell it is. And if you draw little boundaries around the cells, you can see which cells tend to cluster. And then you can make these cluster maps where on each axis you have different cell types. So this is maybe an immune cell, and this is maybe a neuron. So you can see, oh, how often are immune cells close to neurons given by this color, right? So this can tell us overall what kinds of environments exist, which cells tend to cluster. But I think it's just really useful to just look at these maps, and you can see who's next to whom, Learn a lot from that. So hopefully you're convinced that if we can see what kind of cells there are in tissue, we can learn a lot about the biology. Now, how can we do this? Well, MERFISH stands for multiplexed error robust fluorescence in situ hybridization. Basically what this is, it's FISH, fluorescence in situ hybridization, which is a well-established method. We've just figured out a way to make this multiplex, to do this for many, many genes in parallel. And this was published from Xiaowei Zhuang's lab, my postdoc advisor's lab, in 2015, and since then we've improved it a lot, and now it's a commercially available tool. So the way it works is RNA is the product of genes, right? So if you have genes in your cell, and the cell expresses a certain kind of genes, it will make RNA transcripts. So if we can see these transcripts, then we can tell what genes are active in a cell. How can we see the genes, the transcripts? Well, we can label them. So if we design and synthesize oligonucleotides, these are short snippets of single-stranded DNA with fluorescent markers on them. Then we can, if we know the sequence of the different RNAs, then we can target them with these different markers. And so you can do that with different colors for different RNAs, and you can image that. So this has been possible for a while. Rob Singer was the person who first came up with this method, and it's phenomenal. You see these like essentially digital spots where each spot is one RNA, and you can just count them, see how many copies of each RNA you have in each cell. Now, unfortunately, if you use different colors for different RNAs, you'll run out of colors very quickly, or money if you're building a microscope. So we need to figure out a way to do this for more than a couple of RNAs at a time if we want to get a fingerprint of what kind of cells there are. So this was a problem that we and a lot of other people were faced with, and one of the solutions was to just do this sequentially. To hybridize RNA in situ, we need to fix the tissue anyway. So these are cells that are fixed. We can take a section of tissue, we can fix it. It's not going anywhere, so we don't need to label all of them at the same time. So somebody had the brilliant idea to label them one at a time. So we can label one RNA with one set of probes that are fluorescent. We can then remove the fluorescence in some clever way, and then do this for another RNA, and then another RNA. And in this way, we can detect if we do 20 washes, then we can detect 20 different RNAs. But it's very unsatisfying because it's kind of like a linear scaling with time. So we were thinking if we could do this in a better way, and my advisor had this brilliant idea that you can actually look at the same experiment, but as a binary code. So in imaging round 1, we only visualized RNA1. In imaging round 2, we only visualized RNA2. And in imaging round 3, we only visualized RNA3. And so there are some codes that we haven't used. For instance, if there were RNAs that were bright and fluorescent in rounds 1 and 3, we would be able to tell them apart from the other RNAs, right? And so forth. So if we do it in this way, then actually every time we add another round of imaging, we double the number of RNAs that we can tell apart. And so the number of RNAs that we can distinguish goes with 2 to the power of n, where n is the number of imaging rounds. So in 15 rounds of imaging, we could actually distinguish between 32,000 different codes. And this is more than there are genes in most of the cells. So the way we do this in practice is that for each RNA, we first hybridize it with a single-stranded DNA probe. And that DNA probe has binding sites for readout probes, and those are the ones that are fluorescent. So RNA4, for instance, we want it to be bright in round 1 and 3, so it has binding sites for readout probe 1 and 3. So in round 1, we add readout probe 1, it binds here, and then we see a bright spot. Okay, we take note of this. And then in round 2, we wash in readout probe 2. There's no binding spot for it, so it's dark. And then in round 3, we wash in readout probe 3. It's bright again. So this pixel here was bright in round 1 and bright in round 3. We can decode this and tell that it's RNA4. Now we know this is RNA4. So in practice, we do this for many more rounds, and we do this for many more RNAs. But the principle is pretty straightforward. So with this principle, we can now start to look at a lot of genes in parallel in actual cells. So here's published data. Here's a cell in culture. You can see 1 gene, 2 genes, thousands of genes. We can also, because it's all the digital, we can have some codes that are not used, and we can tell exactly how many % of the RNAs we're detecting and how many we're misidentifying because of these barcodes. And then we can do that not just for one cell, but it's just as easy to do it for hundreds of thousands of cells in a dish. And we can do it for tissues as well. With a little bit of machine learning, we could also define the boundaries of these cells and then count the different kinds of genes within each boundary. This will give us a measurement of the transcriptome within that cell, and that can allow us to classify the cell. All right, so so with this, we started to do some imaging in heart tissue. This is from a mouse heart. This is data that was collected 2 weeks ago. Here you see a zoomed in picture of a mouse heart. The white is the boundary stain. This is the cell membrane. These are cardiomyocytes, most likely. The red is the nuclei of cells, and magenta and cyan are 2 different bits. So it's 2 different imaging rounds, right? So what we can tell is that there's a lot of cyan bits here, there's a lot of magenta bits here, probably because this cell is a different kind of cell from this cell. It's too early to tell. So with our microscope, we can actually take many different pictures of the same tissue and then stitch these pictures together. So we can get a high resolution image, not just of a few cells, but we can zoom out and get a better idea of the entire tissue. So I turned off the RNAs now, but you can see the structure. This is the right ventricle of the heart. You can see the orientation of the muscle fibers are different. This is the epicardium, the outside of the heart. Here's the inside. This is left ventricle. This is what fills with the most blood when the heart pumps. You see all these little squares are just individual fields of view. This is all automated. It took about 20 minutes to acquire. The microscope goes frame by frame and just like gets all of these pictures, and we go home and sleep. And then the microscope sends us an email when it's done. So this is great. We can. This is just super preliminary data. We can obviously remove all of these artifacts over time. But what this allows us to do is to really decode the gene expression in the heart. So this is some preliminary data that VizGen got in collaboration with us. So VizGen is this company that makes microscopes, and here you see expression patterns of some of the genes in the heart. So yeah, it's muscle protein, a different muscle protein. Here you see, I actually don't remember what gene that is. This is probably fibroblasts, and here we have immune cells. These are endothelial cells, blood vessel cells. So these are genes that are expressed particularly in certain kind of cell types. But you can put this all together and get an idea of the entire structure of the heart, every single cell, and what it's up to. Not just the types of cells, but also, is the cell healthy? Is it sick? Is it doing okay? Is it communicating actively with neighboring cells? What kind of signaling pathways are active and so forth? So we want to put all this information together and really try to understand how the human heart works, both in health and in disease. And our goal is really to figure out how to keep the heart healthy. We know this is possible because of people like Fauja Singh, who at age 100 ran the London Marathon. So if Fauja can run a marathon at age 100, we can live healthy lives with healthy hearts to at least 80, I would say, right? Right? So there is a solution. We just need to figure out how to keep our heart healthy, what environments are ideal, and what genes are responsible for sickness and disease in different parts of the heart. All right, so I just want to sum up everything we're doing in our lab. It's a combination of DNA engineering, microscopy, and using it to understand biology. So on one hand, we use DNA as a structural tool. We build devices, and we use them to see molecular movements. And on the opposite end of the scale, we use DNA to do computation in situ. I mean, I can go on for days about how cool it is that we're actually reading out bits and communicating with these cells. That allows us to do all sorts of really cool signal processing and error detection that we couldn't do before. But this gives us digital maps of gene expression in situ, and that allows us to understand functioning organs. And everything we do is sort of some riff on this theme where we're trying to be creative with how to use DNA and microscopy to understand biomechanics. I couldn't do any of this without my incredible team. It's just been a year since I started the lab, and I already have phenomenal people. A lot of the hard work was done by Yuening, a postdoc in the lab, and the Origami team is Ryan and Amanda, Lauren, Bella is an undergrad who works with Yuening, RC is a technician, Rupal keeps the lab running, and then we have, oh yeah, Blair here was in Sabrina Leslie's lab, so he's phenomenal. He's an MD PhD now. Claire has left the lab already, and she's gone on to brighter things. And then my collaborators, Peng Yin, Richard Lee, Xiao Wei, and Julio Fernandez. Thank you all for your attention.